All right, moving right along. We just finished talking about isotonic solutions. Again, isotonic is where the concentration inside and outside of the cell are the same, right? So we don't have a net movement of water in or out of the cell. So our example down here at the bottom is when you go into the hospital and they have to put in an IV, it's never pure water. It's always a saline solution, which is going to be equal to the solute concentration outside of your cells and inside of your cells, so we don't get this overall flow, which can damage those cells. Okay. So let's look at hypotonic. Here we have lesser strength. So the outside of the cell has lower concentration than the inside of the cell. So what we see is that because the concentration outside is lower than inside, water is going to try and move into the cell to balance out those concentrations. Now, if these concentrations are too different, so if there's too big of a difference between these, too much water can flow into the cell and it can actually burst that, as we see down here in this image. Okay, that cell gets too big and it bursts. And we see it right here, too much water flows in. Now, this doesn't happen with plant cells because the cell wall gives them enough structure that it keeps those plant cells from bursting. But in animal cells, this can certainly happen. Okay? So if you drink distilled water, that's not good, right? You typically don't see people drinking distilled water because it can have this impact on your cells, because all of that pure water has no solutes in it. And so it's going to rush into your cells and it can damage them. Our last type here is hypertonic, having greater strength. So there are more molecules outside of the cell than there are inside of the cell. And so water is going to rush out of the cell and it's going to kind of shrivel it up. But again, with our plant cells here, that plant cell overall keeps the same structure because of the cell wall. But you can see the inside is in fact shriveled up still. And here we've got this red blood cell really shriveled up because all of the water has left it. So when we talk about people being lost at sea and dying of dehydration, well, there's all that water there. But the issue is that it's salt water. So it's got all of these particles in it that when they drink that water, it forces water to leave our cells and it causes the problem to get worse. Okay, because we need that water in the cell for all of those cell functions. If it leaves, our cells shrivel up and they can't do any functions that require that water. Okay? And so here we see this again. Here we have a red blood cell in an isotonic solution, and we see that they've got that disc shaped exactly as we'd expect to see. Here we have a red blood cell in a hypertonic solution. So water flows out because there are more particles outside of the cell than inside, and they shrivel up like this. And then hypotonic, in this case, water flows in and it causes them to kind of expand. In this case, they aren't exploding or bursting yet. And then here we've got some images of plant cells. Okay, so here we have hypotonic, where it all spreads out because water flowed into the cell. And then here we have hypertonic, water went out of the cell, and so it's all shriveled up. You see the difference in where the plasma membrane is between these two plant cells. Okay. So here we put a cheek cell in a hypotonic solution. Which direction is the net movement of water going to be? Into the cell, right? If it's hypotonic, water is going to move into the cell and that cell is going to expand. What if you put a liver cell in isotonic solution? What's going to happen? Well, there is not going to be a net movement. We're going to have movement out and in, but those are going to be equal to one another, so we have no net movement of water. Okay. Talk about laxatives. Okay. So why do they relieve this constipation? essentially what we see is this milk of magnesia or magnesium salts, which tends to be in a lot of laxatives, is going to put a lot of solutes outside of our cells within our intestines. And so water is going to leave our cells over here and flow into the intramembrane space, making that inside of the intestines more liquidy and allowing you to pass whatever blockage you might have. Okay. Looking at another type of transport here, we have active transport. So passive transport are diffusion, facilitated diffusion, and osmosis did not require energy. Active transport does. And so sometimes we want to move things against the concentration gradient. We want to move things from low to high concentration. And in order to do this, we need energy. Okay? If you think about it, passive transport is like going down the stairs. It's pretty easy to do, okay, or going down a slide. Active transport is like going up the slide. It requires us to exert energy in order to do that. So what we have is we've got various proteins that are going to help us out here. Okay, so we've got some that are pumps, and it's going to require that energy molecule, adenosine triphosphate. We need that ATP in order to move things up that slide. So we're moving from low concentration to high concentration. We've got a couple types of mass active transport where we're going to move a lot of molecules at once. And the first one here is endocytosis. The prefix endo means in, cytosis refers to the cell. So in endocytosis, we're moving things into the cell. So this can be large molecules, it could be a lot of molecules, whatever it happens to be, but we're moving them into the cell. And what we see here is this was a vesicle that is attached to this cell, and it's moving all of these molecules into the cell through this endocytosis. Okay? So we've got a couple of different types here, but you don't need to know those. Just know that endocytosis, we're moving molecules into the cell. This requires energy. The opposite of this, exocytosis. Okay? Exo means out. So we're moving things out of the cell. So this could be waste products that have built up within the cell and we want to get them out. So we bundle them all in a vesicle, take it to the plasma membrane as we see up here, and release them. It could be hormones that are made in the cell. We can release those this way, or other molecules that we make and need to get to other types of cells. Okay, so we create a vacuole. All of those molecules are within it. It fuses with that plasma membrane and then releases all of those molecules. Okay. Now we can jump into our organelles. Okay, so we're getting out of our transport and we're looking at these actual parts within our cells that are going to carry out various functions. Okay, one of the most important ones is the nucleus. Okay, and we've seen it shown very similar to this structure. That nucleus holds all of the DNA within the cell. Okay, a couple of components. We've got the nuclear envelope. Just like we have a cellular envelope, that plasma membrane, we also have something surrounding this nucleus, separating it from the rest of the cell. And this is to protect that DNA. So the plasma membrane is selectively permeable, as is the nuclear envelope, but it's even more so. Not a lot of molecules are able to make their way into and out of the nuclear envelope. Okay. Chromosomes are just DNA. It's a certain form that the DNA is in. The nucleolus is where we're producing ribosomes, which we'll talk about later. And then we've got the nucleoplasm. And this is just kind of that cytosol of the nucleus. Okay, so we'll pick up on the rest of these organelles in the next video.